Hello students, today's our topic is atmospheric layers and microorganisms in air. As we all know, Earth is surrounded by a blanket of gases and we call it as atmosphere. Air is not a natural environment for microorganisms because the physical and chemical conditions that are prevailing in the air will not support the growth and reproduction of microorganisms. Then, how these microbes are present in air? Actually, the microorganisms that are present in air are liberated from various other sources like soil, water, plant and animal surfaces, and human beings. In this video, we will see about different atmospheric layers and the microorganisms that are present in these different layers. The atmosphere consists of 78% of nitrogen, 21% oxygen, 0.9% argon, 0.03% carbon dioxide, and 0.01% hydrogen, traces of other gases, dust, and condensed vapor. In addition to all these, air also contains microorganisms. The study of airborne microorganisms is called aeromicrobiology. Atmospheric layers. Earth is surrounded by the blanket of gases. It has various layers ranging up to the height of 1000 kilometers. These layers include troposphere, stratosphere, mesosphere, thermosphere, and exosphere. Troposphere is the atmospheric layer nearest to the Earth's surface and extends up to a height of about 11 to 18 kilometers. Troposphere contains a heavy load of microbes. However, air is not a natural environment for microorganisms. Physical and chemical parameters of air do not support the growth and multiplication of microorganisms. Microbes present in the troposphere are actually liberated into air from other sources like soil, water, plant and animal surfaces, and human beings. Air acts mainly as a medium for dispersion and transmission of microorganisms. Several infectious diseases are transmitted through air. Several factors affect the distribution of microbes in troposphere. As the height increases, the amount of microorganisms decrease in atmospheric layers. Temperature decreases with increasing height of troposphere. Temperatures at the top layers of troposphere range between minus 43 to minus 83 degrees Celsius, which are below the minimal growth temperatures of microorganisms. As height increases, there is a reduction in atmospheric pressure, concentration of oxygen, and water availability. As height increases, microorganisms are also exposed to high intensity of light radiation, and there will be more exposure to UV radiation that may cause lethal mutation and death of microorganisms. Also, low concentration of organic carbon is found, which will affect the growth of heterotrophs. The physical conditions in high altitude limit even the existence of autotrophic organisms. atmospheric layer above the troposphere is called stratosphere. The stratosphere is characterized by low mixing of gases and contains a layer of high ozone concentration. Ozone layer absorbs the ultraviolet rays of sun and protects the earth's surface from excessive UV radiation. But exposure to high concentrations of ozone and UV radiation are detrimental to microbes. Thus, the stratosphere acts as a barrier to the transport of living microorganisms to or from the troposphere. Coming to microorganisms in air. Microflora in air is less compared to soil and water. Air is mainly acting as the transport or dispersal media for microorganisms. Microorganisms are mainly confined to the troposphere. The microbes in the troposphere include vegetative cells and spores of bacteria, fungi, algae, viruses, and cells of protozoa. As air is exposed to sunlight, it will have higher temperature and less moisture content. Hence, microbes cannot survive in the air for longer periods. Microflora of air can be divided into outdoor microflora and indoor microflora. Air found outside the buildings is called outdoor air, and the air found inside the buildings is called indoor air. Microorganisms present in outdoor air is called outdoor microflora, and microorganisms found in indoor air is called indoor microflora. Dominant microflora in the outdoor air are fungi. Two most common fungi include the Deuteromycete cladosporium and the Basidiomycete yeast Sporobolomyces. Other common fungal forms include the Basidiospores, Ascospores, Conidiospores, and the mycelial fragments. Bacterial forms are also found in outside air. The common forms include the spore-forming species like Bacillus, Clostridium, and non-spore formers like Sarcina lutea, Micrococcus luteus, and non-pathogenic species of Corynebacterium and coliforms. Air microflora over the sea surfaces are quite different from that of landscapes and includes organisms like Achromobacter, Bacillus, Corynebacterium, Flavobacterium, Micrococcus, Sarcina, and Staphylococcus. Microorganisms are detected up to very high altitudes of the troposphere. However, spores are dominant than vegetative forms at high altitudes and can be seen up to a height of 30 kilometers but in low concentration. Coming to factors affecting outdoor microflora. Microflora of outdoor air is not constant. Several factors affect the distribution of outdoor microflora. As height increases, microbes are subjected to low temperature, UV radiation, and desiccation. Number of microbes varies from place to place due to differences in human population densities. More microbes are found in crowded areas and also in air over terrestrial surfaces than over the sea. Variations in the spore densities in outdoor air occur on an annual and daily basis. Annual variations are due to climatic changes, mainly because of the changes in temperature. Very high temperature and low winter temperature are less favorable for microorganisms. Also, during the rainy season, microbial spore density in outdoor air is less as rain washes out spores in the air. Diurnal fluctuation in spore liberation also affects outdoor microflora. For example, colorless basidiospores are liberated usually during night. Sporangia of phytophthora are liberated in the morning when the relative humidity is low. And also, the spores of Ericife are released during midday. Coming to indoor microflora and factors affecting indoor microflora. Air inside the buildings is called indoor air and the microorganisms in it are called indoor microflora. Indoor microflora is not constant and is affected by air circulation through doors, windows, and due to turbulence caused by moving people. In indoor air also, fungi are the dominant microbial forms. Saprophytic fungi like Penicillium and Aspergillus being the most common ones. Bacteria in indoor air is determined by factors like ventilation and the number of occupants in the room. Common bacterial forms include Bacillus, Staphylococci, and Clostridium perfringens. Indoor bacteria is mainly from human skin and respiratory tracts. Pathogenic bacteria or viruses can be seen in the indoor air if the occupants are infected.